Hi everybody, and welcome to the mini lecture on carbohydrates. Be sure you have something to take notes with. Go ahead and pause here and grab it if you don't. This information will be very useful. We can consider this a pre-lab. The next few mini lectures are building up to your lab for this week. Before we jump into carbohydrates, let's just track back a little bit and talk about the four major classes of macromolecules. So you have your carbohydrates, which is the focus of this mini lecture, proteins like amino acids, lipids, which are fats, and nucleic acids, which are RNA and DNA. So the thing that all macromolecules have in common is that they're all organic, and you'll remember that organic means made of carbon. They all have a carbon backbone. And also, just an interesting tidbit, the four major classes of macromolecules make up most of the dry cell mass. So if we were to weigh a cell, most of the things that have weight in the cell would be a carbohydrate, a protein, a lipid, or nucleic acid. So they are very important to the function of cells. So carbohydrate, you guys know I love these Latin words. So carbohydrate comes from the Latin root carbo for carbon and hydrate for water. So carbohydrate means made of carbon and water. And there are three classes of carbohydrates: monosaccharides, disaccharides, and polysaccharides. So let's look at the monosaccharides first. Monosaccharides are made of one carbohydrate monomer. So we have in this example a glucose monomer. Other well-known carbohydrates are fructose, ribose, and maltose. And if you have a pack of gum or some candy hanging around, if you look at the ingredients, it will probably have fructose, ribose, glucose, or maltose as an ingredient. These are simple sugars, very sweet. So glucose, speaking of, is probably the most important macromolecule, certainly the most important energy molecule. So it's an important, not the only, but certainly an important energy source for living things. If you remember from General Biology 1, plants make glucose through photosynthesis, and then humans and other animals eat plants to consume this glucose. So this is a major source of our own energy. The second type of carbohydrate is disaccharide. So disaccharides are made of two monomers by dehydration synthesis. Remember that? So in dehydration synthesis, if you remember, the hydroxyl group of one of the glucose and the hydrogen from the fructose. So we have these are our two monomers. This hydroxyl group is attracted to this hydrogen. They break off and leave, forming water. That's the dehydration part. And then the bonds left open by the leaving of the hydroxyl and the hydrogen form together through this glycosidic bond. And then we have a sucrose which is made of one glucose monomer and one fructose monomer. So this is the synthesis. Dehydration, loss of water, synthesis, new molecule being made. Sucrose is table sugar, by the way. And then the third category is polysaccharides. So polysaccharides are made of long chains of carbohydrate monomers. They can be branched like this top example, amylose here, or they can be, they can be unbranched like amylose we see here, or branched like amylopectin that we see down here. Starch, cellulose, which is an important component of plant cell walls. Cellulose gives a plant its particular shape. So starch, cellulose, and chitin. Chitin is the thing that bug exoskeletons are made of. So it's the reason that bugs crunch when you step on them. So starch, cellulose, and chitin are some famous polysaccharides. Also, plants store sugars in the form of starch by stringing together long chains of glucose. So as a plant is making glucose through photosynthesis, any glucose that it doesn't immediately use will be attached to another glucose by dehydration synthesis, forming a long chain like this amylose that you see here, storing this energy. And when the plant needs the energy, enzymes will break these bonds, and as we talked about in the previous section, we talked about biosynthesis of macromolecules. The breaking of bonds releases energy. So these enzymes will break this storage version of the sugar, which is a starch, down into glucose monomers, and then the plant can use those or the human, whatever, can use those monomers for energy. And we'll talk a little bit more about enzymes and the role they play in altering reactions when we get to the protein section. Okay, so in the lab this week, we will test some unknown substances for the presence of macromolecules. There are specific tests that help identify each type of macromolecule present, and this is a lab that we really would have done in person had we been there, but luckily the internet is coming through for us, so we'll have a virtual lab for this. Now we can test for the presence of monosaccharides by using Benedict's reagent. This test is sometimes used to test for the presence of glucose or sugar in the urine, which is an early warning of diabetes. So sometimes if you've ever gone to get a physical or checkup and they have you pee in a cup, they're going to take that and submit it to a Benedict's test to see if there's sugar in the urine. All right, so how does Benedict's test work? So you take your sample, you add Benedict's reagent, you boil it for about 5 minutes, and then you record the result. And what you might see is one of these. So when we mix sugars with Benedict's, it causes a reaction and the Benedict solution changes color from blue to dark green or greenish yellow if there's a little bit of sugar available, orange if there's a moderate amount of sugars in the sample, or brown or dark red if there's a large amount of sugars in the sample. Now the Benedict's test only works for simple carbohydrates, so only those monosaccharides, a couple disaccharides, but not many. So remember in a minute, because there's going to be a question on the next slide, that Benedict's test only works for simple carbohydrates. That means the carbohydrates that have been reduced to their smallest, the monomer form. Okay, so now let's think about it a little bit. Would a cracker, which is made of carbohydrates, just a regular old kitchen cracker, a Ritz or a saltine, would a cracker give a positive result in a Benedict's test? Let's see, what did you think? No. So crackers are made of complex carbohydrates. That's why they're a no-no for people who are watching their diet. So crackers are made of complex carbohydrates. So the carbohydrates that make the cracker would need to be broken down to its smaller components, right, to its monomers, in order to have a positive result in a Benedict's test. So a cracker is made up of those polysaccharides. So it would not give a positive result in a Benedict's test. If we did a Benedict's test, the result would still be blue. Okay, so let's try another question. What if you chewed the cracker first? Then would it give a positive result in a Benedict's test? What did you think? So the answer is probably. Okay, so the digestive enzymes in your saliva would definitely break down some of those complex carbohydrates in the cracker. So you probably get an intermediate result like that greenish yellow or orange, yellow-orange color. Okay. All right, great. So go back if you if you need to watch again to take some good notes.
So that'll do it for this mini lecture on carbohydrates. So you can move right on to the next
proteins.
macromolecule, which is lipids. See you there.